Good afternoon, everyone. Can you see my slides and hear me okay?
Yes.
Yes. Okay.
Yep, looks good.
Thank you. So thanks for joining this episode, I guess, of the monthly CCEH Flow Friday Seminar Series. My name is Katie Siu. I'm an assistant professor in the Division of Experimental Hematology and Cancer Biology at Cincinnati Children's Hospital. And today I will be talking about the applications of imaging flow cytometry, specifically to the study of erythropoiesis, but hopefully you will find it broadly applicable to other parts of hematopoiesis. So just to give you an overview of what I will talk about, I will give you an overview of erythropoiesis because I recognize this is not something everyone that studies hematopoiesis is familiar with, and some of the analysis challenges that we encounter. Then I will introduce imaging flow cytometry and then describe a couple of different applications in which researchers have used IFC to study erythropoiesis and the erythropoietic niche, including highlights from the literature as well as some of my own research in collaboration with Dr. Theodosia Kalfa. So this is a very brief outline of hematopoiesis. I'm sure you will all recognize how very much brief it is and overly simplified probably, but this was sufficient to kind of introduce the topic of erythropoiesis and that we're talking about the production of erythrocytes or red blood cells. And I know a good amount of hematology researchers, especially in our division, are mostly focused on the hematopoietic stem and progenitor cells. And the myeloid cells, because of their obviously important roles in immune regulation and hematologic malignancies and things like that. And in fact, their first step in a lot of hematopoietic stem progenitor cell analyses is to lyse the red blood cells. So today, hopefully, we can all learn a little bit about erythropoiesis. So I wanted to share this fun fact that it's estimated that the body produces about 2.5 million red blood cells per second in order to maintain homeostasis. And so as important as all those immune regulation and malignancies are, this is also a very important field to study because if you cannot maintain that level of output of red blood cells, you obviously will die. So this is an overview of erythropoiesis, which of course begins with a hematopoietic stem cell and progressively committed progenitors that undergo self-renewal up into a point in which they enter terminal erythroid maturation. Now, these self-renewing erythroid progenitors, the burst-forming and colony-forming units erythroid, have recently had a flow cytographic method described for analyzing them. I'm not going to go into it here, but I've included the reference here if that's a population that you're interested in analyzing. Our research and interests focus mostly on terminal erythroid maturation, which begins with the first morphologically identifiable erythroid precursor, the proerythroblast. And from this step on, the erythroblasts undergo a series of progressive changes, undergoing one cell division per stage effectively, and drastically decreasing their cell size, their nuclear size, condensing their nucleus a lot, downregulating lots of genes that are not essential for their erythroid functions, and ultimately ending by extruding their nucleus to become a reticulocyte in the peripheral blood. These reticulocytes, once they enter the circulation, mature over about 2 to 3 days to get rid of any remaining organelles and intracellular contents, as well as reduce down their membrane size and finally mature into a fully mature biconcave red blood cell. So underneath, I've shown what happens to a few key surface markers that are often used to identify either stem and progenitor cells or more mature lineage committed erythroid cells. And one of the things that you can see is that there's not really other than like the specific expression of TER119 and the characteristically high expression of CD71, which is the transferrin receptor, there's not really a lot of unique immunophenotypic markers that permit evaluation of these individual stages of terminal erythropoiesis. So they pretty much all have TER119 except for the very earliest proerythroblast, and they basically all have some level of CD71. So this is great if you're like pulling them out in a lineage dump because you get them all with TER119 mature all the way up to basophilic basically. But if you want to study processes that are happening in terminal erythropoiesis, it's helpful to be able to identify the different stages. And so even though this is an imaging flow cytometry talk, I wanted to start by touching on methods that we already have using traditional flow cytometry to identify terminal erythroid precursors. And so this has obviously relied heavily on the presence of these markers CD71 and TER119 that I mentioned. And so a commonly used one that we see a lot in the literature still is the use of CD71 and TER119, where the TER119 low cells are attributed to the proerythroblaст population, and then everything else that's TER119 positive can get broken out maybe by size, since we know that that's an obvious feature of their maturation. And this was based initially on a protocol for fetal definitive erythropoiesis, which was described by Harvey Lodish's lab, and then Merev Sokolovsky further elaborated it. But this seems to work really nicely in the fetal liver, but it doesn't really seem to bear out in adult definitive erythropoiesis. And what I've tried to show here is that even when you separate these by size, these things based on their CD71 expression, they kind of overlap a lot. And other than the ones that are completely CD71 negative, which are your mature red blood cells at this point, all the other erythroblasts kind of overlay each other. And this point was really made very well by Mohandas Narla's group in New York in an earlier PNAS paper in 2009, and then further in 2013, where it was refined further. But basically, they compared the expression of CD71 and CD44 over terminal maturation and found that CD44 actually declines at a much more perceptible rate across these populations, allowing a better resolution of separation. Interested in the granularity of the different erythroblast populations. The CD71 approach is certainly sufficient to say these are erythroblasts. I would just make sure to caution people against claiming that anything based on CD71 expression represents a specific population. We've definitely seen papers where they gate in this top cluster up here and call them basophilic erythroblasts, and that's certainly not the case. These, when sorted out according to this paper, you can see very clearly that you do get sort of a progressive maturation here, and most importantly, want to also note that this final population that kind of roughly corresponds, if I added CD71 here, corresponds also to the CD71 negative population, that these are really red blood cells, and they're probably coming mostly from breakage of the vasculature when you're flushing the bone marrow or crushing the bone marrow or whatever, and they're not, shouldn't be counted when you're evaluating and quantifying erythroblast populations. That should be restricted to the nucleated cells that are up here in these first 5 populations. So with that said, this is sort of the point that I'm going to lead into with the imaging flow cytometry, is that, we still don't have any other markers. We had to, even to optimize this flow cytometry analysis, we had to harness their change in size, which is a characteristic morphological change that these cells undergo. And so we were only able, you can see here, that even just based on CD44 expression, these cells don't separate out. They have to use the forward scatter to see the change in the cell size. And so these morphological features dominate in identifying these different cell types, which are clearly morphologically distinguishable, but in flow cytometry can be tricky to kind of separate out. Before I move on, I just want to touch on that this has been worked out for human erythropoiesis as well. A good amount of what's in my talk is based on murine erythropoiesis, but I wanted to throw this in as well, just in case that's what your main focus is, that there have been several papers published also by Mohan's group that identified optimal ways to identify different progenitor and precursor populations in human bone marrow and human erythroid cell cultures. So why do we need to refine these populations better or use a different method to study terminal erythropoiesis? Well, there's a lot of specialized processes that I think are unique to erythropoiesis, and this could be applied to other cells as well, but specifically within erythropoiesis, there's a lot of processes that are very stage specific. So genetic regulation that's happening during that terminal maturation process where one cell's not quite turned things on yet, and then you go to the next stage and suddenly a whole different program is running. Enucleation, which happens at the very end. These cells associate cell clusters with macrophages within erythroblastic islands, and that's something that's been of great interest lately in the field. There's pathological shape changes, so again, this morphology aspect that you can't really capture with flow cytometry, and that's relevant mostly in sickle cell anemia, but probably in other pathological conditions as well. I've seen uses where they look at it, they use it to look at parasite infections with Plasmodium. So there's a lot of things that you can do with imaging flow cytometry to study these things. And the reason that we need to do that instead of using traditional methods like flow cytometry or microscopy alone is that, you know, we have, like I just described, this continuum of differentiation marked by these very different changes in RNA and protein expression. So you can't just do a bulk analysis because there's all these different heterogeneous types of cells in there. There's also a lack of distinct markers that allow us to separate out these different stages easily by cell sorting. You have things like rare events, rare populations. The proerythroblasts are the least in number, or like in enucleating cells where this is sort of a small but important population to evaluate. And, you know, once you begin terminal maturation, these are cells that are not self-renewing, so you can't maintain them in culture. You have very limited numbers. You can maybe keep them a day or two in culture. But if you want to study something that's happening in terminal maturation, you need to be able to analyze the cells quickly and in bulk without a lot of sorting and manipulation. And so that's where flow cytometry doesn't quite cut it, but microscopy can be very tedious because you spend the whole time looking around on the slide to figure out, you know, where an orthochromatic erythroblast is. So the solution that we've found for our research has been the imaging flow cytometry is a really good, happy medium for this. So you get the morphological detail from microscopy and the high throughput multi-parameter analysis that you get from flow cytometry. So if you're not familiar, an imaging flow cytometer basically takes the PMTs that you would use to detect your fluorescence in flow cytometry, and it replaces it with a CCD camera that you can take fluorescence images on. So you're simultaneously doing light and fluorescence microscopy as well as this high throughput interrogation of the cells and their fluorescence and their characteristics that allows for the multi-parameter analysis that you do afterwards. So it's really the best of both worlds. The resolution isn't as good as microscopy, but it's way high throughput, and vice versa. You don't get the imaging with the flow cytometry, but you get all these parameters as well that you can analyze. And seeing really is believing. So this is really the best of both worlds. And I think there are many imaging flow cytometry options available now. The one that I've included here and what most of my experience is with the Amnis image stream, which we are now on the model Mark IIX. This is now recently acquired by Cytech, but this was originally created by Amnis and is the one that we have available here at Cincinnati Children's and the one that I see published the most often, I think, being used, but there are definitely other options I think available from other manufacturers now. So this is what the analysis sort of looks like, just to give you a really brief idea. If you've never seen it before, you have your analysis area where you have your flow cytograms that you're used to seeing with flow cytometry, and histograms of whatever parameters that you find meaningful for the analysis you want to do. This includes things like the distribution of the intensity of a certain fluorochrome, but also like the area, because you've got a two-dimensional image that you can also take measurements on. You can do the aspect ratio, you can do mean pixel intensity, circularity of a fluorescence image, compactness. It's really quite endless and very customizable to the research question that you have. On the left is the image gallery, where you can see the corresponding bright field and fluorescence images of each and every cell that you've recorded. So every dot, of course, in your flow cytograms corresponds to a cell. And here you can actually see what that cell looks like and where that signal is coming from. So I think Jim Pallas's group, led mainly by work by Kathleen McGrath, was the first to publish an erythroblast analysis scheme utilizing the image stream. And they leveraged this very powerful and customizable multi-parameter analysis to create and define parameters that were very meaningful to the evaluation of different erythroblast stages. And so that's not like the one-size-fits-all of flow cytometry, where you're basically going based off of intensity of different markers. But here with the image stream, you're able to do more morphology-based features, and they relied heavily on presence of DNA and RNA content, the nuclear size, the overall cell membrane size, and I guess a surface marker or two in the TIR119 and the c-kit to identify all of these different erythroblast stages, but mainly utilizing morphology characteristics. And I added the imaging analysis, or the gating analysis, here just because it's a flow cytometry talk, and I figured I should have some plots as well as the pretty pictures. But you can read more about it here. And this has actually been further refined and worked on over the years. And so there's an updated methods paper on identifying the different populations of erythropoiesis in the image stream, if that's something that you're interested in doing. And you can see here, and they've included even an analysis of the pyrenocyte, which is the nucleus that gets extruded on the basis of its low TIR119 expression and retention of CD71. So you can expand your analyses to looking at things that partition with the pyrenocyte, perhaps in sort of a more high throughput manner. So this method was then used by other researchers, including the Kalfa lab, and they further exploited it once they were able to use this to identify a population of orthochromatic erythroblasts, and then further interrogated that population in order to identify cells that were undergoing enucleation. So this is a rare event, very important to study though, and very challenging to isolate otherwise and maintain intact. But by being able to just fix and load the entire cell population in the imaging flow cytometer, you can preserve these events and then pull them out on the basis of their features. And so to do this, they, you can see here, utilized the aspect ratio, which is the elongatedness of the cells, the major to the minor axis, and then developed this custom parameter they called the delta centroid. And that's basically the distance between the center of the stain for the nucleus and the center for the stain for the membrane, based on the TIR119 or whatever membrane marker you have would work. And so this allowed them to identify and find a population that was enriched in cells undergoing enucleation. And then once you have found that population, then you can interrogate that specifically for the markers that you're looking for. So in this case, they were looking at cytoskeletal organization during erythroblast nucleation. And you can see these actin and tubulin structures that form during the process, and the nuclear symmetry, asymmetry that typically preceded enucleation, was found to be reduced as measured here when you inhibit microtubule assembly with colchicine. So all of these measurements are able to be done with the analysis software from the ideas from the data that you get from the image stream. The thing is that you have to know, one caveat I would say is that you have to know what you're looking for. So to be able to customize and define these features can sometimes be a little bit daunting and limiting because the possibilities are really endless. Other applications I just wanted to include in case this is something that you're interested in working with peripheral blood is there's been, I think, attempts to use imaging flow cytometry to identify sickled red blood cells, as well as pathological cell aggregates as a result of the stickiness of red blood cells in sickle cell anemia. And so there's some references here if that's something that you're interested in learning more about. I wanted to include a vignette about my own personal experience using the image stream to evaluate a novel case of a syndromic type of CDA in several unrelated patients, and how we were able to utilize imaging flow cytometry to do some of this rare event kind of analysis that was essential to determining that this mutation was causative for the patient's phenotype. And so this was, very briefly, a story of 3 unrelated patients that had a syndrome of kind of CDA-like disease with transfusion-dependent anemia, binucleated erythroblasts in the bone marrow, as well as erythroblasts joined by these cytoplasmic bridges that you can see here. And the whole exome sequencing was performed after an initial sequencing was done, and they didn't have any mutations in any of the known CDA-causing genes. So the whole exome sequencing revealed de novo heterozygous mutations in a gene called VPS4A. And this is a component of the escort3 machinery, and it's been implicated to be required for a variety of cellular processes, including the abscission step of cytokinesis, as well as endolysosomal trafficking. And so we used the image stream to evaluate the localization of VPS4A during erythroblast cell division using cultured human CD34 positive cells initially. And we found that, as had been reported for HeLa cells and such in the literature, in the dividing erythroblasts, the VPS4A localized to the centrosomes at the beginning of cell division, and then migrated to the spindle poles, and then ultimately ends up migrating to the midbody for the abscission step of cytokinesis. And this is where it is required to perform that final step. And so we repeated these analyses using iPSCs from normal healthy donors and our patients. And again, these are very challenging experiments to do, since the erythroblasts are on a one-way street to differentiation, and they don't really handle synchronization well. They grow in suspension, so it's not like you can grow them and have them adherent and just fix them in place and image them. So these are kind of challenging things to do. So we were able to, just fix the cells directly, stain them in suspension, and run them in the image stream. And because of the high throughput capabilities, we can still find parameters to pull these rare events out. And so in this case, I was using the aspect ratio of the nuclear stain and the area of the nuclear stain to identify these cells that had basically 2 nuclei that were kind of far apart from each other. And you can see very clearly the tubulin-based midbody here in the center, and VPS4 right in the middle. However, in the patient lines, VPS4 never managed to make it into the midbody, got kind of close, but it never really got to the center where it needs to make its final cut. And we quantified this in a number of dividing cells that we caught. And we also identified many cells that had lacked tubulin in the midbody structure, but still retained, based on the GPA staining, a membrane connection. And this was very reminiscent of the cytoplasmic bridges that we saw in the patient's bone marrow. And so we did look at these more closely using confocal microscopy, because that is one caveat of the image stream, is you're not going to get, confocal level resolution, but it's a trade-off for the high throughput capabilities. But as you can see, these are definitely cytoplasmic bridges without any DNA or tubulin in them, just like we observed in the patient's bone marrow aspirates. So theoretically, when these eventually coalesce back together, if they don't rupture or break spontaneously on their own, they're going to generate a binucleated cell, which is likely why our patients had increased binucleated cells, and we were able to quantify these in our cultures as well using the image stream analysis. So one other part of this was because of the role of VPS4 in endolysosomal trafficking, we were interested in evaluating the behavior of CD71, which is the transferrin receptor, and the reticulocyte maturation, because this is thought to occur through this similar pathway. So when we normally evaluate reticulocytes using flow cytometry, we do this using thiazole orange and CD71, and freshly released reticulocytes from the bone marrow will be very high for their CD71 still, and they will also still contain RNA as well as some other things, which the thiazole orange will stain for the RNA. So over the 2 to 3 days in the circulation that they are maturing, the reticulocytes will lose their CD71 expression through loss of their membrane and through release of exosomes, potentially, and the thiazole orange will also drop then after that, and they will become double negative red blood cells. In our patients, however, with these VPS4A mutations, you can see that the pattern is really disturbed. There's a lack of continuity from the high early reticulocytes to the mature red blood cells, and there's this new population down here that's CD71 positive but TO negative. And so we were very curious what these could be. You really don't know. One potential explanation could have been that they are lysed cells that don't retain their TO or their RNA anymore, but still have CD71 on the membrane. However, this was not the case, so we put them in the image stream so that we could find out. And you can see in the CD71 double positive, CD71 TO double positive population, these cells look very much like reticulocytes, and again, you have this benefit of being able to see what the cells look like. So these look like reticulocytes. They have kind of larger, floppier, messier membranes. They haven't matured. They contain a lot of TO staining within their cytoplasm, and they have CD71. Down here, we have the CD71 positive TO negative population, and these, as you can see, are very clearly intact cells with CD71 retained on their membranes, and they have more matured membranes. They have smaller membranes with starting to have like a biconcave disc shape. So these are more matured cells, suggesting this is maybe an alternate maturation path for them, and some sort of difficulty in clearing the CD71 from their surface. We also then went ahead and used the image stream to evaluate transferrin uptake in reticulocytes from these patients. And so this you could quantify with image stream, I guess, or with flow cytometry, I should say. But using the image stream, you're also able to count the spots. So you would just get like a single intensity measurement and say, okay, there was less. But here we can actually do a spot count and say like what the average median number of spots per cell is. And this is for the control compared to the patient. And so we found that they take up less transferrin despite having an excess of the receptor, suggesting that there is some sort of block in the endolysosomal pathway. So switching gears a little bit from single erythroblast analyses to the erythroblastic niche in the bone marrow. All of this maturation of the terminal erythroid precursors is occurring within these niches in the bone marrow called erythroblastic islands. And these are specialized macrophages, presumably, that the erythroblasts associate with, as well as the surrounding erythroid and myeloid cells. And the macrophages are known to play an important role in phagocytosing the extruded nuclei. And there's also been some evidence and suggestion that they could provide iron and growth signals during erythroid maturation. However, this last point especially is really not well understood yet, and there's a lack of evidence for this, mainly because we've long been unable to identify and isolate these specific erythroblastic island macrophages for study. And there's been a huge push in the past 5 to 10 years to really understand the identity and the immunophenotype of the erythroblastic island macrophage so that we can understand how they contribute to and support erythropoiesis. And so this is another the very basic, overly simplified at this point, overview, but just to introduce a couple of markers. But we had some very basic understanding of adhesive interactions that regulate the attachment of erythroblasts to the macrophages. And this is sort of what we built our expectations of the immunophenotype for them on, right? So they had long been studied and identified using F480, which is considered sort of a pan macrophage marker, but we understand now is very promiscuous and maybe not so pan for macrophages, but when the research started, this was the gold standard, was that they had F480. BCAM1 has been shown through some nice experiments to play an important role in mediating an interaction with alpha-4 beta-1 integrin on the erythroblasts. And so BCAM1 was always known that they had to have BCAM1. There's some other markers that have been proposed and have more evidence now since this was made. Maybe we can take the question marks off now. But these were some of the markers that people were looking for when we first started down this road. And so when there was this big push about 10 years ago to figure out the identity of the erythroblastic island macrophage, some groups started looking at this in sort of novel ways. And they started trying to use traditional flow cytometry, but kind of non-traditionally by looking not at the single cells, but at the multiplets. And so by looking in the cell clusters area, and then looking at cells double positive for F480 and TER119, they concluded that this population of cell clusters that had both erythroid and macrophage markers were erythroblastic islands. Another group, Levesque's team, published an even more elaborate immunophenotyping scheme where they did a similar approach though and used the large cell clusters rather than the single cells, and a marker for TER119 again for the erythroids, and this time CD11B, with the assumption that the macrophages being monocytic derived would have CD11B on them, I guess. And then this combination of markers, F480, BCAM1, CD169, goes on, and claimed that, you know, they had identified this immunophenotype of the erythroblastic island macrophages. So, of course, this is cytometry. It's intended to be a single cell method because you can't be really sure what you're looking at when you're looking in the cell clusters. And, you know, so are these even really EBIs? And what if all erythroblastic island macrophages are not the same? What if it's not just a monolith, but what if there's heterogeneity based on the needs of the different erythroblast populations or something? So we took a different approach and we decided to try using the imaging flow cytometer to analyze erythroblastic islands from the bone marrow. So after optimizing the collection of intact EBIs from the bone marrow and doing otherwise a similar approach of looking at the cell clusters and then at the events that were double positive for both an erythroid marker and a macrophage marker, we then, within this gate though, could actually look at the images and interrogate them for what really was the main defining characteristic of an erythroblastic island macrophage, which was that it had CD71 positive erythroblasts surrounding it. And so that morphological aspect was really important for identifying these as erythroblastic islands. And we were able to do this in loads and loads of EBIs, and now had a tool to characterize the markers on the erythroblastic island macrophages and the different features of them. So one important point is that this was absolutely essential, and it's a cautionary tale, I guess, for these noble and novel attempts to use traditional flow cytometry to look at cell clusters, but that not everything in this gate was a true EBI. And you can see some examples here that I pulled out of just random cell clumps, looser clusters that are not as tightly associated as these grape-like overlapping clusters that we get for the EBIs. And then down here, another important aspect is that there is now our lab and others now have reported this, that fragmentation of the macrophages leads to a loss of some membrane remnants on both CD71 positive cells and CD11B positive cells for us. And that this has obvious ramifications in the attempts to isolate and identify and define and characterize, even at a single cell level, the macrophages that are involved in these islands and maybe other macrophage populations. But obviously, even sorting on single cells, you can't trust your F480 positive population as just F480 positive cells when you've got large populations of cells that contain small bits of F480 positive macrophage membrane. So this is an important thing to keep in mind, and a good reason to always, if you have access to an image stream, maybe just once put your flow panel in the image stream once, just to see if the populations you're gating on are what you think they are. So once we were able to identify the macrophages, or the UBIs and their macrophages, using the image stream, we were now able to interrogate for these immunophenotypic markers, but in a much more robust way. So looking at VCAM1 and utilizing some of the features in the ideas analysis software, you can do a similarity, which is basically creates a Pearson correlation coefficient for two markers. If you want to objectively say that these two things are co-expressed, this is probably a good way to do it. And so this showed that there was a high correlation between VCAM1 staining and the F480 staining. For CD169, the results were a little more mixed, and that was because we saw two different types of staining. We saw cells that had a lot of CD169, cells that had more punctate and really dim CD169. So if you were gating, you might not always included these ones that had more like small puncta of CD169. And that could affect analyses later, but by being able to visualize them and still compare the puncta that we saw with the F480, we were able to conclude that most of the macrophages did have CD169. There's just this difference in patterning maybe, or level of expression. And then finally, the CD11B that was used by the Levesque group, we found that that was not correlated well with the F480 expression. However, we did find CD11B within these cell clusters, and surprisingly, it was just not on the macrophages, but in cells that were on the clusters. And so this launched a whole nother area of research, and what we found was that when we looked more closely, approximately 75% of erythroblastic islands actually had CD11B positive cells within them as well. And you can see that here. And so we also looked in situ to make sure that this was not some sort of artifact of our preps, but in fact, in intact bone marrow, you can find islands where there's a macrophage wrapping its little cytoplasmic tendrils all over both CD11B positive cells and CD71 positive cells. So what we were able to do using the image stream again, because we have these area and morphology features, is by measuring the area of CD11B and the area of CD71 around these islands, we get this sort of scatter plot. And now each one of these dots represents an erythroblastic island, and the the positioning of it on this graph tells you how much CD71 and or CD11B it has. So a dot down here is a pure erythroblastic island with just CD71 and no CD11B, and a dot up here has almost half and half. And this ratio, if you take do a linear regression and kind of get the slope of this line, it ended up being a really consistent number for multiple preps, suggesting that this is really like a physiological balance. And not only that this is a real interaction, but that this is something that maybe we can use as a readout of the proportions of the cells in the islands. And so we did a lot of work that I'm not going to discuss here, but basically these CD11B positive cells we determined were granulocytic precursors, and they are residing within the islands and maturing alongside the erythroblasts potentially. So we did some experiments looking at things that would perturb granulopoiesis to see what would happen to this balance. And so again, this is all quantification of data collected on the image stream, though, where we're using that CD11B and CD71 area as a readout of how much of each cell type is in the islands. And so when we stimulate with GCSF, we see this ratio go up to having more CD11B in the islands and less CD71. And the opposite effect, using a genetic model, the GFI1 knockout in collaboration with Lee Grimes, this has a defect in terminal granulopoiesis, and so we see the opposite direction where we see a shift towards more CD71 in the islands and fewer CD11B positive cells. So next, we targeted the other lineage within this niche, the erythroblasts, by stimulating the mice with EPO. And you can see that not only do we get the expected shift towards more CD71 positive cells in the islands, we actually saw a huge increase in the number of EBIs. And this was really interesting because that means either the EBI macrophages have to proliferate or there's recruitment of monocytes to support the increased number of islands and the increased demand for erythropoiesis. And so we took these EBIs from both the saline and the EPO-treated mice and enriched their erythroblastic islands and did a single-cell RNA-seq experiment. And you can see there's a lot of populations that came out of this, including many different monocytic populations and the granulocytes that we expect and the erythroids that we expect to be there. But only one population changed substantially in response to EPO, and that was this macrophage cluster 9. And so this now gave us a tool to go in and look and see what other things these erythroblastic island macrophages are expressing. And using our transcriptomic data and a variety of other things, we identified that the erythroblastic island macrophages very specifically express SPICE. So we were already in the process of collaborating with Jason Gardner at the University of Cincinnati, who was also looking at SPICE in a different context, but we had these SPICE mice along with Jason. We looked at their erythroblastic islands, and sure enough, the SPICE GFP labels almost 100% of the erythroblastic islands in the bone marrow of the mice. And so this is quantified here. And not only is it in most of the EBI macrophages, but it's relatively low expressed across the bone marrow. It's less than 1% of the total bone marrow are SPICE positive cells, and it's a really small percentage of the total F480 positive cells. So this could be a very sensitive and specific marker for the erythroblastic island macrophages. So that concludes my vignettes about the applications of imaging flow cytometry to study erythropoiesis. Just to summarize, the high throughput and multi-parameter analysis that you get from imaging flow cytometry gives you the best of both worlds. You have all the best features of cytometry and microscopy. The analysis of terminal erythropoiesis can be aided by imaging flow cytometry in a variety of ways, but mostly identifying the specific stages of erythroblasts, either for rare events and to make sure you're directly comparing cells that are at similar stages, since their features and their functions and characteristics are changing as they are. And so you can also use this to pull out a particular erythroblast stage for further analysis, like the orthochromatic erythroblasts for enucleation, or for something else that is a erythroblast stage specific process. Imaging flow cytometry also permits high throughput analysis of erythroblastic islands or other cell clusters that you might want to apply it to. And the best feature of this is not only just seeing it, but then your ability to create customized masks to analyze and compare just specific components. So I know I'm getting a little long, so I didn't spend a lot of time on the spicy, but you can actually create masks to restrict the analysis just to within a certain area. And so this is showing the intensity inside the macrophage F480 stain, so that you're not like getting signal from other cells that are like stuck or whatever. You can look at that also, but the ability to be able to say, I only want to quantify GFP inside the macrophage is really helpful. So that's another key feature and concept. And then, you know, a general cautionary note that care should be taken with erythroblastic islands research and any associated macrophages, just knowing now about the fragmentation thing. And this goes for anything. And again, I think I already said it once, but I would urge you, if you're already doing something by traditional flow, if you haven't put it in an imaging flow cytometer once, just to see if your populations are as clean as you think, I would. And finally, before I conclude, I just want to say that one caveat of all of this is the parameterization. So the great thing about imaging flow cytometry is it's very customizable, but you have to know what you want out of the analysis, which can be really overwhelming, and it can also just be really challenging. So it takes a lot of intuitiveness and kind of messing with it a lot to even think about what all the possibilities are that you could put together to create custom parameters and stuff. And so something that's really exciting for future outlook is that they've now released a machine learning software with the ideas analysis. And they actually have an even more expensive like AI version, but the machine learning one, I think, is probably good enough for most of your research needs. But the machine learning algorithm allows you to simply input what you think are the features that will define or segregate the cell populations you're interested in. So very broadly, like shape, CD71 intensity. You know, you just kind of tell it sort of broad strokes what you think will help, and the machine learning will go through and come up with parameters and features that are very customized, and actually combine them and come up with ways to separate out the two cell populations that you're interested in separating out. And I think that this is really going to expand the applicability of the imaging flow cytometry to different areas of research, as well as make it more accessible to researchers who maybe have a perfect test case for what it should be used for, but maybe don't know where to begin with the analysis or don't know what features to use to do their analysis. So the machine learning algorithm, I think, is really going to take this to the next level. So with that, I just want to thank you all for listening, and I'm happy to answer any questions now, or if you want to send me any email or get more information about any of the literature or the experiments that I discussed. And I, of course, want to thank our Research Flow Cytometry Core for the image stream and maintaining it, and our Center of Excellence in Hematology, and our funding, and all of our collaborators for the work that I presented. Thank you. Are there any questions?
Hi, Katie.
Hi, Jim.
How you doing?
Good, how are you?
Good. That was very nice. Very, very nice. I was just wondering whether you have a feel as to the, I believe if I recall, the islands you've really been focused on has been in the bone marrow. And, these islands show up also in the spleen and in the fetal liver. I was wondering whether you have a feel as to uh, whether these are going to end up eventually showing up as very different sounding or looking populations, or they're going to be very similar, or do you have a feel?
Um, we did some comparisons, preliminary comparisons in the first paper in Frontiers in 2017 when we first described this method. And it did seem like these had different sort of properties overall,
Mm.
especially between the fetal liver and the bone marrow in terms of just general shape and things. Like there were a lot of things that I wanted to explore in the data that I wasn't able to, but I think we mentioned the circularity feature was showing something different in the macrophages. I think that was in the paper. But yeah, like the macrophages just sort of look different, and they did have some differences in markers, specifically like between the spleen and the fetal liver. Also, I think we don't see CD11B positive cells, which I think is expected, that you don't see a lot of
Oh,
myelopoiesis in
yeah.
those organs. And so I think that the macrophages may very well be different in that their functions are distinct and that they're solely supporting erythropoiesis, and maybe the fetal liver ones may even come from a different progenitor. So
Uh-huh.
I think I would expect. And so as you've looked very carefully at the fetal liver macrophages, I think you've probably noticed some differences in what we've seen in the bone marrow compared to the fetal liver, right? So
Yeah. And I guess relatedly, I just don't know whether, has anybody, maybe you have looked at stress after stress, whether um, are there more erythroblastic islands, or I mean, or does that change during stressful conditions, or
in the spleen or in the bone marrow?
I guess anywhere, maybe in the spleen, since that seems to be.
So in the spleen, we don't usually see erythroblastic islands at baseline.
Oh yeah, I remember you. I think you told me
There's,
that once. Yeah.
yeah, there's not usually any islands at baseline, which is, I guess, not surprising, but also interesting since you have sort of the red macrophages there and they're doing their job. They're eating
Sure.
the red cells, but they're not supporting erythropoiesis, and maybe because the stress progenitors aren't there. But yeah, we don't see islands at baseline, just under stress. And in the bone marrow, we haven't done a lot of other conditions, but just like with the EPO, they do expand. So you do see an increase in number in the bone marrow with, at least with EPO. We haven't done other
Like
modes of stress yet.
phenylhydrazine treatment, would that cause a
We haven't done a lot with phenylhydrazine. We've done phlebotomy, and
ah,
phlebotomy will induce increases in splenic and medullary erythroblastic islands.
let me see. Thanks.
Mhm. Right. Well, guess if there are no more questions, no one minds if we sign off early. Thank you all for listening, and feel free to contact me if you have any other questions about imaging flow cytometry.